Hello, thank you for tuning into this streaming webinar, Lightning Fast QC with NIR. My name is Ryan Palermo, and I am a market manager and former product manager and product specialist for the BUCHI benchtop NIR products. Today, I'd like to speak briefly to the capabilities of our BUCHI FT-NIR within pharmaceutical quality control programs. First, I'd like to describe an all too common scenario. You have a high volume of materials earmarked for quality control on one side, whether incoming raw materials, in process, or finished products, and on the other side you have limited resources by which to accomplish that workload. This combination of factors can easily manifest in inefficiencies and lost profitability. For example, the need to quarantine materials until they can be released, and the delay of manufacturing until confirmatory quality tests are received. Moreover, when resources are tight, the sampling capacity is typically reduced. This is counterproductive to improving product and process understanding. NIR, or near-infrared spectroscopy, is a tool that can help avoid those QC lab bottlenecks. So what is NIR? NIR is a technique that uses low-energy light, typically on the order of 800 to 2500 nanometer wavelength, to measure material properties like identity or composition. The measurement principle is based on the fact that molecules vibrate. When the vibration of the molecule matches that of the incident light, light is absorbed, giving us a characteristic spectrum. Particles also scatter light in unique ways, providing evidence of physical characteristics to the signal and further enriching the spectral fingerprint. Chemometrics is the way that we can relate that spectrum and its information to the qualities of interest, generating our reports. So how can NIR be of use? Here's a short list of key benefits. First, NIR is fast. Measurements are typically on the order of 30 seconds or less, much faster than a traditional wet chemistry method, allowing you to avoid long hold times on QC samples. Secondly, NIR is non-destructive. That means that no sample is consumed as a part of the method. There is no dilution, no combustion, etc. The sample used for an NIR measurement can be used for another QC method if needed or held on retain. Avoiding dilution also means no chemicals. This reduces user health hazards as well as environmental impact and reduces waste management costs for the lab. Lastly, the NIR method is simple. Once an application is set up, a user needs only to enter the sample name into the software, then prepare the sample for measurement, whether that means pouring the sample in a cup or placing a probe in contact with the sample material, then clicking a button to initiate the measurement. A straightforward OK or not OK result is shown. No special chemistry or computing skills are required for routine operation. Here is an overview of NIR applications within the pharmaceutical and other industries. NIR is most widely used for raw material identification and 100% label inspection. There are also quantitative applications that apply to raw material receiving, including compositional analysis like moisture, protein, or fat content. In-process or intermediate analysis is also possible. Typical examples include things like blend uniformity or determining percent moisture in a drying operation. Finished product testing using NIR can be accomplished on powders, liquids, or compacted solids. Examples also exist of identity or counterfeit testing on finished products. The foundation for success of any NIR method is a sound physiochemical basis of the measurement. NIR active vibrational modes are limited to mostly carbon-hydrogen, carbon-oxygen, oxygen-hydrogen, nitrogen-hydrogen, and sulfur-hydrogen bonds. Water is a particularly good absorber. So, materials which lack these types of bonds are unlikely to be a great fit for this technique. However, there's a caveat. Since NIR signal is the result of both absorption and light scattering, non-organic compounds may sometimes also be evaluated with the technique, as well as things like particle size or density. Implementation of any NIR method should follow a careful risk assessment and validation procedure to ensure absolute suitability. Instrumentation is the second level to evaluate when considering suitability for an application. All NIR instrumentation will not perform equivalently. Some instruments, particularly handheld spectrometers, suffer from a limited wavelength range or signal-to-noise ratio, which may limit performance. There are also differentiations in things like light throughput and signal-to-noise ratio. High light throughput allows for a greater volume of sample to be interrogated by the NIR light. This provides more representative measurement results. High light throughput also enables successful measurements for very turbid samples or for measurements in a transmission geometry, something important when evaluating a metric such as content uniformity. Other factors like wavelength precision and stability and regulatory compliance of hardware and software should be considered. In terms of sampling, there are often various sampling geometries available for NIR instruments, optimized for liquid, solid, or semi-solid handling. The third level of our hierarchy is chemometrics. Chemometrics is the science of extracting information from chemical systems by data-driven means. It's how we relate the spectra we collect to the quality attributes we desire to calibrate for, things like the identity of a material or the percent of an active ingredient. The most important aspects of the calibration development is going to be the sample selection. As with most things in life, the adage garbage in equals garbage out applies. Sample selection is then followed by application of appropriate chemometric algorithms and, of course, internal and external validation procedures. Our BUCHI NIRware software offers a powerful calibration wizard and reporting tools to help guide that process. Please reference some generic applicability of NIR for the pharmaceutical industry in the USP document chapter 1119. NIR is also an iterative process. You begin by defining the scope of the project. What do you want to accomplish and with what setup? Then define elements impacting the spectral response for your project. Things like sample temperature, seasonal variation, processing equipment, etc. This step also includes verification of analytical performance of the NIR instrumentation itself. Next comes data collection, establishing those data for which to train the calibration and which data to test the calibration performance. The identification or composition of calibration and validation samples should be verified by traditional compendial reference methods prior to use in an NIR model. This is followed by calibration, then validation of the model and the instrumentation. The life cycle of a calibration does not end with the start of routine use. Performance of the method should be monitored routinely and updated with samples that demonstrate new sources of spectral variation, maybe things like particle size or compositional variations as they occur. When new samples are identified to augment an existing calibration, you run through the loop once again. This is what the application development cycle looks like in our NIRware software. We have three unique modules: NIRware Management Console, NIRware Operator, and NIRCal, which interact with the Microsoft SQL database. Nearware Management Console is where applications are created, spectral properties and results are managed, and where security settings are defined, among other administrative tools. Nearware Operator is used to operate the instrumentation. NIRCal is the chemometric software used to create and/or test calibration models. Once a calibration is developed, it is appended to an application for routine use in the operator software. There are some example qualitative applications published by BUCHI, including identification of raw materials, identification of real versus counterfeit tablets, and blend homogeneity monitoring. Please reach out to us if you want a copy of any of these application documents. We also offer extensive training on how to accomplish these tasks in-house. Here are some general guidelines for developing qualitative applications. First, please consider that spectra and samples from at least 5 unique batches or lots of material should be collected for use in method development. Those lots should span as much of your anticipated sources of spectral variation as possible, whether that's multiple suppliers, variations in moisture content, particle size, or some other variable. Those lots will then be partitioned into either a calibration or validation set. It is also good to keep in mind that an NIR library of tens to hundreds or even thousands of materials is built in segments. Developing a calibration from small groups of chemically similar materials helps to optimize the calibration performance, especially in terms of its discriminating power between like substances. It also helps to avoid false positives. BUCHI does offer a so-called pre-calibration for pharmaceutical compounds. This developed library includes spectra and calibrations for 68 different materials, including 15 active ingredients, 41 excipients, and 12 liquids. The spectra of these materials have been matched to quality certifications and/or compendial method approvals and can be used to jumpstart your own calibration or extend your current calibrations. I mentioned a few times the potential to do quantitative applications as well. Here's one example pulled from a Best at BUCHI publication for API determination in tablets. In this case, calibration samples included tablets of 2 different drug loadings, represented by both pilot and production scale manufacturing. Pilot scale samples ranged from 75 to 125% label claim. 2 different tablet presses were used for production scale samples and another for pilot scale. For calibration work, HPLC was used as the reference for API composition. All sources of known spectral variants were considered in the sampling plan, including the positioning of scored versus unscored tablets, and by varying the tablet press used, which impacted tablet hardness. The calibration results for the 25 milligram formulation are provided in the table and graph here. Metrics such as precision, bias, regression coefficient, Q value, and consistency were evaluated for both the calibration and validation set samples. The comparability of the calibration and validation errors, SEC and SEP, indicate good method robustness. The bias value shows good correlation between HPLC and NIR predictions. An additional test set of 6 samples was used for external validation. The table here shows the results of those 6 tablets of nominal drug loading using the NIR and HPLC methods respectively. The deviation is small, with a mean value of deviation less than 1%. This indicates good suitability of the NIR method for the task. With that, I would like to thank you for watching this webinar and invite you to direct any comments or questions to our ussales@buchi.com email address. You can also find additional product information and resources for NIR and other QC laboratory equipment like Keldol, flash chromatography, or melting point on our webpage at www.buchi.com.